got a great introduction. I don't need to go on. Just I'm the guy who could never figure out what he wanted to be when he grew up. So I did a lot of different things. There's probably one of your favorite companies that I actually worked for in there. People ask us sometimes why we actually renamed our track. So it started off as biotechnology and bioinformatics. And I pushed for a couple of years. I said, we really need to call this something different. Let's call it digital biology. And the question we get is, well, what is digital biology? What does that mean? And so instead of giving something like some extensional definition with a lot of examples, I want to give one example that I think is really telling. I want to tell you the story of this guy. This is Santiago Ramon y Cajal, right? Nice, good, traditional patronymic Spanish name. He was a famous scientist back in the day, back in the old world, and he actually won the Nobel Prize for medicine back in 1906. And you would think he would be happier looking, being that guy. But he's not, and part of the reason is he's sitting in the wooden chair that he sat in about 16 hours a day, hunched over a microscope, that microscope there, a variety of microscopes, because he was studying neuroscience the old way, observational biology way, where he would peer in and look at brain tissue. And actually, because of a new stain that was developed and what they were able to do, he was the first person on Earth who actually realized that brains are not just a big clump of cells shoved together, but they're networks, that the cells actually connect to each other. Because that wasn't obvious just looking at it, or just looking at it with a microscope. You had to have some other technology there too. And so he then painstakingly drew these networks that he saw, and his stuff is still used today in modern medicine textbooks. And that's pretty amazing. But that's also old world, and I mean that with respect, but that's the old school, old world way to do it. What's the new world way to do it? Well, if you want to understand how brains work, you find something with the smallest possible brain possible and take it apart. And here's this wonderful model organism, C. elegans. It's a worm, and we know that it has exactly 959 cells. Seriously, that's every one of these that are female. The males have less, which is scientific proof that men really are less complex than women, at least for these worms. And they really do have exactly that many cells, and exactly 302 of them, whenever they are mature, are neurons. Not 302, 303 or 301, but 302. And they form exactly 6,393 connections. And we have that entire thing mapped out. And because of that, we can actually digitally model and simulate most of the things about the worm, but for sure the worm's nervous system, its entire connectome. We can build a model of it, and we can actually go ahead and run that model digitally on a computer in a digitally simulated environment with physics that work kind of like the worm's muck, and we can see what happens. We can take this organism, and this is the first organism on Earth. We've mapped all the stuff and then woken it up inside a computer. This is what it looks like to wake up in the Matrix. Check this out. This is the digital simulation of that worm, with its connectome running some body muscles, and it's moving around a digitally simulated environment. And it does what that worm does you saw on the other slide. It's looking for food and sex and avoiding toxins, basically. It's not, it's not exciting. It's not a night out in San Francisco, but it's swimming around. And this is a pretty good simulation, and they're actually working on making this better and better. The guys who are working on this are the Open Worm Foundation. It's a .org. And they are really seriously building the world's first digital life forms now with an S, and doing it open source. And all the stuff that they work on, they upload to GitHub. And people contribute to the different connectomes, environments, et cetera. And they're doing a lot of genomics pieces. They're doing a lot of pieces on how it works, how it interacts. And in fact, to give you an idea, because this is open source, somebody said, hey, I could take that brain network for a worm and not wake it up in a digital simulation. I could wake it up in a robot, a Lego robot. This is what that looks like. So this is, keep in mind, this is not programmed. This is not like a Roomba running into things. This is a worm's brain running this really simple robot. And again, it's looking for food and sex. And it's going around and it's finding obstacles and backing up. And it was a fairly simple task to port this over to this. And so, worm first. They're working on some more complicated insects. They've got plans for the mouse, and then eventually, as Ray Kurzweil will tell you, humans. So this is what we mean when we talk about digital biology and we talk about the new world and the old world. And I wanted to give you guys a flavor for that. So let's go on in the little bit of time I got left and talk about some of the things that I usually talk about to give folks who've never seen this before some grounding in what's going on in digital biology today. This is what I do. This is what my days are made up of. I'm a DNA guy. I like to sequence DNA. I like to read it. I like to go ahead and do some genetic engineering. And I like to make new tools that enable all of this stuff to be done cheaper and in places where it hasn't been done before. 10 times to 100 times to 1,000 times cheaper is really our goal for this. This isn't the way I think about what I do. If I had a business card that had one company on it, it would probably say, Raymond McCauley, the 3 R's of DNA hacking, reading, writing, and hacking DNA. This is what I do, and this is what I want to tell you guys a little bit about. So let's go ahead and start with reading DNA, kind of how that works. One thing a lot of people are now getting exposure to is 23andMe. How many people here have done 23andMe, have heard of it? Excellent. For those of you who haven't, these guys took a cancer research chip that you can use to tell what one individual's DNA, how it's different from the person sitting next to him. You can go ahead and compare those two by running their samples on one of these chips. It takes a special reader, some other things. 23andMe said, let's go ahead and release this to consumers. We won't do cancer research with it. We'll let anybody who cares to go ahead and take this test, and we'll put their results up on a website that only they can see. And they can go ahead and tell some stuff about what's going on. These are the kind of things that it shows you. Do you have any genetic conditions, congenital diseases, things that you would inherit or be a carrier of? Do you have any genetic risk factors for chronic diseases like cardiovascular disease, Alzheimer's, other pieces, cancer? Do you have a different response maybe than your neighbor to drugs? Drugs are sort of prescribed now to this average person, but we all have really different responses to drugs, partly because there was no million years of evolution to curb how our responses to drugs were. So some people need higher or lower dosages, or drugs just don't work at all. And some of the stuff, what they call the fun genetic traits, like can you taste bitter things? Is your earwax flaky or slimy? What color are your eyes? Of course, you can look in the mirror for that, but you can go ahead and predict it genetically. What are your blood groups? Are you lactose intolerant? And that was what they were selling this as. They were saying, hey, let's do this as recreational genomics. The FDA didn't like that. They said, when you tell somebody they're going to get cancer, that's not recreational. They kind of shut it down because of some of the things that they were releasing about what's going on with people's genetics. And so they had a little set to, and 23andMe is still sort of recovering from that. Just in the last month or two, they came up with a new model where they're not showing as much of this stuff. What they are showing, and what's kind of turned out to be their killer app, is who are you and where are you from? Who are your people? This is actually a small bit of mine. Most of my stuff, there's maybe 300 different races and subraces that they track. This is most of my stuff up here in the top. I'm boringly Northern European. But I've got a couple of interesting things in there. And I found out about some of this. There was a funny thing that happened to me a year or two ago. One of the things you can do with 23andMe is whenever they run your sample, they will actually tell you, hey, this is what we found out about you. Would you like to share that with other people who are also running their samples? And you can opt in. You can say yes. You don't have to. They don't do it by default. It's not Facebook. So. And then they'll go ahead every night and run a job that compares every new sample to everyone who's already in their database, which is close to about 1.5 million people now. And so I will get these little emails that say, hey, Raymond, we found a new fifth cousin for you. But if you know a fifth cousin, man, I mean, I've probably got about 3 or 4 fifth cousins in this room, so that's not that big a deal. But I got one, hey, Raymond, we found a new second cousin. I was like, yeah, wait a second, second cousin. I was like, didn't write him a birthday card, don't know who that, oh, I know all my second cousins. And I looked it up and I said, yeah, I want to communicate with this person. And they communicated with me. It's a young woman, young African-American woman in Dallas, Texas. I'm from Texas originally. And it turns out so was my uncle, who was a traveling salesman in Dallas. And she's like, this is so exciting. I never knew who my father was, and to be able to communicate with my family. I'm like, I got to tell you, you weren't missing much. Sorry. He's dead. I can talk about him. But it was really interesting. We had this conversation like, should we show up at each other's Christmas dinners and freak out our families? Or what do we want to do? And so we still talk a little bit today. It's neat. It's interesting. But it shows really that this kind of data is not just scientific data or big data or genetic data. It's personal data. It's about who you are and who you're related to and who those people are in your life. And every time you share your stuff, you're sharing half of the data of your parents, siblings, and kids. And so you might think about it. And we're going to see more and more of this. So this is what we do whenever we really want to look at your data. We don't run 23andMe. We don't run one of these little chips. We actually sequence your DNA. And it's a complicated process. And this is where I have lived and worked and eaten for a couple of decades now. And I love this, and I could tell you about it, but not in 30 minutes. So we do this whole thing, and the interesting part for you guys to know about it is we do it really, really cheaply. And in fact, the price for doing this has gone down precipitously. This is one of those nice semi-log graphs that we're in love with at Singularity University that shows every major break is a 10x improvement in the price performance. So we can see the price going down pretty quickly for this at about a Moore's Law rate. And then at Illumina, a little company called Solexa, we got acquired. We invented this new thing called next generation DNA sequencing. And all of a sudden, year after year, the price of DNA sequencing started going down at a rate of 5x a year. And nobody had ever seen anything like that. Literally, nobody had ever seen anything like that in any engineering endeavor that humans have done. And we didn't really believe it would keep up from year to year. But more or less, with a few business reasons why it hasn't when people are protecting profit margins, more or less it has. So let me give you another way of looking at what that means. If you look at, we completed the Human Genome Project, 2001. All in that was $3 billion. $3 billion to sequence basically the first human genome. $2 billion will buy you a US B-2 stealth bomber. So you've got enough left over to make best friends with everybody in the world who wants to go to the opening weekend of Star Wars 7, right? And you've still got enough left over to take them all to Jurassic World and to the Avengers, and the good one, the first one, not the one with Ultron. So expensive is the point. By 2007, we had gotten it down to $1 million to sequence a human genome. A company called 454 sequenced one of the guys who had discovered the structure of DNA. Jim Watson sequenced his genome for $1 million. That'll buy you a mansion in upstate New York, or currently about a 500-foot walk up in downtown San Francisco. So. But a lot of money still. You don't want to put it on your credit card. 2013, we got it down to the price of a used car. This is one out of actually East Palo Alto Craigslist. Air conditioning doesn't work, a little hubcap issue, but it's a great automobile. You'll be fine. Now, I had been predicting for 9 years prior to this that we would be able by the end of 2015 to hit a $1,000 genome. I actually almost really closely got fired for this a couple of times. The company I was working for was like, don't tell people that. One, they think you're crazy, and two, they were going to wait 9 years to buy our thing, you know? And it turned out I was wrong. It wasn't by the end of 2015. It was in January of 2015. We hit the $1,000 human genome. That makes it comparable to the price of a chest x-ray, at least in the US medical system. So nice medically relevant bit. Now, if you keep looking at this, this is where we go next. 2018, we're going to get it down to the price of a delivery pizza. And probably it'll be 30 minutes where it's free. 2022, I'm hedging a little bit on this, but I'm still predicting we're going to have it cheaper than the price of flushing a toilet. That the water in that toilet will cost more than it costs to sequence a human genome. In California, it'll cost a lot more. Right? And that's going to be really interesting whenever we get to that point, because we'll be sequencing genomes all the time, including the genomes in your toilet. That toilet will be the most wired appliance that you've got in the whole house. And we'll sequence your cells and the cells of things in you and the cells of the things that you ate. And there'll be a little report that goes out in email, and you'll get the call, and it's like, really? Are you still eating the chorizo? Because we talked about this. You can't do that anymore. And you'll be like, oh, Raymond McCauley, and pulling the cables out of your toilet. This is where we're going with this, and this is happening really fast. And this matters really deeply to people who are doing research in disease mechanisms, including all kinds of cancer, and who are looking at all sorts of pieces, so geeks like me, but also I think it matters to you guys. This is the so what about this. What does this mean to you? In the very near future, medicine stops being reactive, where you get something and you try to figure it out and treat you and get you back, roll you back to a healthy state and do something about that. And it starts being more predictive, where we have an idea of what you're going to get. And in fact, we'll have an idea, I think, this is how it will work. A baby is born, and you do that little heel stick and you get that drop of blood. Right now, you do that at Stanford, and they test for 20 diseases. We'll actually do genetic tests. We'll just run the whole genome. We'll put it in an electronic health record that works, and we'll read that off. And in fact, a little AI will go through and say, here are the things for this pediatrician to worry about in 0 to 3 days of this child's life. This is the punch list. And then 0 to 3 months, 0 to 3 years, because there's lots of infant mortality stuff that's genetically determined. And then it's going to kind of slow down. It'll be, here's stuff that you're going to want to know about your adolescent. Here's stuff that we won't tell the parents, but an 18-year-old human being with full rights about determinations what they want to do with their health, we'll let them know, because maybe you've got predilections for diseases and you want to do something about it. You want to change a lifestyle habit so that you're less likely to get it. Or failing that, because as human beings, we're not always good at doing that. Maybe it'll be, hey, we're going to figure out, maybe you need to come in for that first colonoscopy not at 50, but at 30, because we think that's going to save this many hundreds of thousands of lives in the medical system. And we will do that, and this will just be the way medicine works. To give you an example, and it's already kind of old now, we've seen this happen. People don't get amniocentesis anymore. They get these non-invasive prenatal tests, where instead of sticking a needle in the belly, and that's hard. In fact, how many people here have ever had to go through amnio? A few people? I salute you, brave gentlemen, raising your hands. I know that amnio was hard. That was tough. I'm sure your wives held your hands, and poor widow bunny. So, I raised my hand. I had twins with my partner, my ex, and held her hand while we did that. And you know, you get that wince whenever they do the needle in the belly. And then they did the second one for the second twin, and she already knew how much it was going to hurt, and I was trying to make her laugh. They're like, don't make her laugh, for God's sakes, because the needle will jiggle and hit one of the babies. Something will happen. There's a 1 to 2% chance of a miscarriage event from doing this, but you still want to do it. But now we don't have to do it at all, because we've got this test, and all it does is it looks in mom's bloodstream and finds the free-floating DNA that's not hers, that is from the baby, that's from that embryo, that fetus. And you can analyze that free-floating DNA and say, genetic irregularities or not, and decide to do a follow-up. So this has already become a big deal. 80% of amniocentesis has been replaced by this test. We think that we're going to be able to do the same thing with cancer. So if a fetus releases some DNA, then a cancer tumor trying to establish itself does the same thing. You can go ahead and find this DNA in the bloodstream and catch it and say, hey, there's a tumor starting. Let's do something, and not find it stage 4 or 5, find it stage 2 or 1 or stage 0. Stage 0 is going to be what we call it when we find evidence that there's cancer going on, but it's too small to be imaged. Any other way would not detect it. And this is what's going to happen. You're going to get a call. It's going to be like, Raymond, you know, we had some bad news. Don't freak out, but it turns out you got cancer, that test last week. It's going to be okay. We're going to give you 4 pills. You're going to take them this week, and you might get a little sick. Probably not. Most people react well. We'll scan you again in a month. Won't that be a nice future to live in? The prediction is by 2020, we're going to have the 30 main tests that you want to be able to test for. And they will replace doing a mammogram. They will replace doing a colonoscopy. You won't have to go to the crusher anymore. That's what my ex calls it. That actually does you more harm than good. The radiation from that, cumulative over a lifetime, may be doing you more harm than good. Colonoscopy, it doesn't do you more harm than good, but you don't want to schedule more than one of those. You do your first one, you're like, I'm fine. So we'll have something else. Now keep going and getting those until you know. Right now we've already got the first one of those 30 tests released. It's for a nice form of colorectal cancer. They can do an early determination of it, but we've got to fill in a lot of blanks before you stop going and getting these tests. We've got a lot of people working on this. You don't have to have cancer for this to work. You can go ahead and determine if you've got a familial risk for cancer. The guys at Myriad Genetics used to do this. It was 10,000, then it was 5,000, then it was 4,000. Now it's 250 or 199. You can put it on your credit card. You don't have to worry if insurance is going to cover it. You can find out if you were at risk for this Angelina Jolie type cancer. This is the kind of testing she did through Myriad to determine if she needed to take action. So this is where we're at. And there are a lot of companies following suit. A lot of people are working on this. I'm going to skip this part. I like to talk about life insurance, what's going on, but life insurance is cracking down on people who've had genetic testing. I do want to just bring up that there are a lot of companies now working on cancer DNA testing, treatment, detection, screening, the whole thing. And at the very end, if they don't run me off stage, I'll have a slide with some interesting companies to look at. It doesn't have to always be about humans. It doesn't have to always be about medicine. We can use the same technology to look at our food. So the whole idea, hot dogs. What are hot dogs made of? Genetically test them. Who likes hot dogs? I'm so sorry. I'm going to show you this. Let me show you 3 quick things. 2 of them are real serious. One of them is just kind of, everybody's going to go, oh, and we'll talk about it. So the first thing is, these guys used genetic testing to test hot dogs, and they found that about 15% of the hot dogs they tested were problematic. Meaning the all-beef hot dogs, not so much, right? There was chicken, something else stuck in there. They found also, whenever they looked at protein content, somebody's like 9 grams of protein per serving. Nope, maybe 4.5. It was never more than, it was always less. Here was the second thing, and I thought this was a big deal. Vegetarian. 10% of those, not so much. You know, when you eat one of those vegetarian things, you're like, wow, this is really surprisingly good. 10%. Now, if you're an ethical vegetarian, hey, you still get points for trying, but if you're somebody with a food allergy or some really serious thing, then 10%, and that's a big deal. Even if it's traces, that's a big deal. Okay, here's the third thing. Take a look at this. Right? Oh. Everybody looks at this and they say, oh, you know what happened? Raymond was in the butcher shop working late, and he's doing the thing, and he's so sleepy, and then he and falls in, and the grinder's going, and they get in the next morning. Salim opens the door. He's like, where's Raymond? Man, I don't know, but look at all the work he did. This is amazing. Let's make some hot dogs. What's really going on here, though, is not that it's a hygiene issue. It's Raymond's working late, and some little hair comes off his head. There's a little sliver of fingernail. There's some skin that gets in the game, you know, when you're chopping things up, which is bad enough. And now we've got this new microscope, basically, this new method to look at these things. So it's interesting what happens and what this is going to do for the food industry. So let me end up this piece and then rush through a couple of other things to say. Next generation sequencing, it's a big deal. It's going to be a bigger deal because we're coming up with even newer technologies to do this. This is my favorite one. I actually have this sequencer, and I had it in my pocket this weekend. I was going to show it to you guys, and I left it in my kid's backpack. And this is the future we live in. I left my DNA sequencer in my kid's backpack. But you can actually take this and use this anywhere in the world, plug it into a laptop. That's all you need to do. I've used it on a rubber raft in the Amazon. I've used it in villages to do diagnostics for Chagas disease. You can just do it. And this replaces roomfuls of equipment. Really quickly, let me run through a couple things with genetic engineering, which is now starting to look more like software engineering, because you can go ahead and decide you want to do a project where you're going to transplant genes from a jellyfish into bacteria to make them glow. And one of our researchers did this in the lab. They designed it up, got something they liked, cut and pasted that into this website, DNA 2.0. They hit add to cart for about 13 cents a base. They got this DNA sent to them by FedEx 2 days later. and they used it to transform bacteria and make it glow in the dark. Now, 20 years ago, 30 years ago, this was a Nobel Prize. Last year, this was a science project for a second grader in Mountain View. A little 8-year-old girl did this, and her dad was not a biotech guy. So she was doing this on her own, interviewing people, finding out what to do, and was successful. And didn't win the science fair, because that's what it's like to live with all the Google people in Mountain View. What we're going to use this for first is industrial microbiology. We're going to go ahead and change bacteria, yeast, algae, and make them make different things for us to replace big, complicated manufacturing processes. The way that this work is, and some people call it synthetic biology. I like industrial biology better. You go ahead and you have these cells are your factories. Your DNA is the blueprint. And then whenever you need to expand industrial capacity, you just make more cells. And that's an exponential process. E. coli bacteria will divide every 20 minutes. So in a long afternoon, you can go from 1 cell to 1 billion cells and keep going. This is already starting to do price and supply chain disruption in a lot of different industries. To give one example, this is what the future of biofuels look like. It's not going to be sugarcane, it's not going to be corn, it's going to be algae in bio-solar panels. This is gene tweaked, it produces a little biodiesel precursor. You just go ahead and change the spigot down in the bottom and drain off what you need. It's a drop-in fuel, basically. We're also able to do things like grow a radio in a petri dish. So not just make bulk materials, but make really complicated stuff. And here somebody did exactly that and then hooked it up to speakers, batteries, wires, and turned it on, and it worked. But it only played country music, so we put bleach on it immediately. I mean, Lyle Lovett's okay, but you got to have some limits. So this is where we're at. We're able to do more and more complicated things. We're changing food, putting nutrients and drugs into food whenever we want to do that to reach different populations. And we're even getting to the point where we're starting to change human genes. Here's my list, my hit list of genes that I'm going to knock out if I have a daughter next. And this is what we're going to do to give her immunities and superpowers. And I would love to talk to you guys about this because I'm really serious about this. The big deal about this, there are a lot of ethical concerns, and there's no way we can talk about them in the whole 20 seconds I've devoted to this in my 30-minute presentation. But I'd like to point out, I think for me, the biggest, most important thing is going to be, what does the economic gap look like whenever you have rich people who can go ahead and change the genetics of their children so they live longer, don't get sick, and are smarter, and poor people who are like, really? And I think the big thing is, let's go ahead and make this available to as many people as possible on a voluntary basis as quickly as possible. But I'm sure half of you are horrified to hear me say that. So we've got a lot of talking to do. And also this will make abortion look like a little bitty controversy in a teacup. So longevity, I'm going to skip this part because I wanted to go ahead and show you guys some other stuff. I'll give you one thing I'll tell you. Longevity is not by your good habits. This is the longest living human that we've ever verified, Jean Calment. She did quit smoking at 119. She started again at 121. But we're finding the genetics of these people who live a long time is an interesting thing. And forgive me for skipping this. I would love to talk to you guys. 3 of the technologies that are going to be really big are stem cells, and all of the different places we can apply that. What we're able to do with DNA nanorobots and other kinds of DNA repair. And then one of the things that came out of Singularity that I'm particularly proud of, the Organ Preservation Alliance. About 40% of deaths can apparently be prevented if you could just have an organ transplant. And if you have to go ahead and get that from a fresh corpse, that's tough. This is what we're going to do as a stopgap until we know how to grow our own organs. So let me go on very quickly to the last thing, which is biohacking. Biohacking is something that means a lot to me, and for me, that is lowering the price of entry into this whole system. We founded, me and 5 other people founded BioCurious. This is the world's first biohacker space. It's basically a gym membership for science. We put a lab together, we stuck it in Sunnyvale, we put $3 million worth of donated and repaired equipment in there, and we charged people $100 a month to access it. And folks came in who were students, teachers, activists, inventors, hobbyists, pro-am people, and all of them were using it. This is what it looked like on its opening day. 2 of the people there are now at Singularity University. This is a class that we taught with some Singularity University people where people were extracting their DNA and analyzing it, building the tools to actually do that. This young woman was looking at her breast cancer risk and her genetics. We're seeing a lot of people use this space and other spaces like it to invent things. Everything from drugs and therapies to different kinds of technical pieces. And we take the risks associated with this very seriously. We worked with 7 different levels of government so we could determine that we could open this without actually doing anything illegal. Who do you think the hardest ones to work with were? Anybody? Anybody? Small business owner? I heard one person say the fire department was the hardest one to work with. They were just like, you can't put that many people in a lab and do this in your exits. The FBI was by far the easiest to work with. They wanted the idea of this being transparent, people coming and going and working on things and talking about it. They actually co-sponsored the world's first biohacker conference with us back in 2012. We had 20 spaces from around the world come to there. Here are some of the spaces that I know of that have just been developed in the last couple of years. We're seeing a tremendous number. While you're here in San Francisco, I always like to point out the closest one is IndieBio. Not really a biohacker space, although it's got its roots there. It's an accelerator for technology. You can walk there from here, but you might want to call first. They do tours, but they like to arrange it ahead of time. And they're actually closing a new accelerator class coming up here at September 1st. There's some other ones in the Bay Area, Counterculture Labs, Berkeley Biolabs, some other places. And I want to point out, it's really interesting what's going on. Miraculous, which is one of the wonderful companies that came out of Singularity U, did its first work in biohacker spaces, did some incredible things. They are on the road to using microRNA as a diagnostic. They have just announced some breakthroughs that they're able to do in detecting stomach cancer, better than anyone has ever done with anything before. And they're making instruments that make this all possible. Let me show you really quickly a little 20-second video. This is what it looks like to do microfluidics with their new thing. See those drops, those dots? They're using electrical fields to move those around, take little samples. It's like Pac-Man. And they need to mix these up. Ordinarily, this would be a biochemist, a PhD, standing with a little eyedropper, basically. And it just goes that quick. These guys are going to be a big deal. And they are one of many people who have started from now modest means, biohacker spaces. Here are just a list of some Singularity companies out of the last 3 years. And this is not an exhaustive list. I'm sorry for you guys that are not on the list. This is what I was putting together very quickly and talked to some of you with permission to put on. They're going to be a big deal. And I would love to talk to you about each and every one of these. Here is my list of companies to watch. This is not my list of companies to invest in. Some of these are going to fail spectacularly, but in really interesting ways. And so you're going to want to see how that happens. But take a look at this. This is a new version. I've done versions of this throughout the years. I appreciate you guys letting me do this and talk about what we do in these garage labs. We're trying to start a scientific revolution. More tools and more hands. We'd love you to be a part of it. If you've got a kid between 5 and 25, go to that link, and there's some resources there for them. If you want to talk to me, don't email me, but get me on the phone. Thank you guys so much.